Today, we sit on the precipice of a new era of space exploration, fueled by the ingenuity of scientists with the yearning to reach the stars. But our fate is not certain, and for the first time in human history, we have the ability to ensure our survival by becoming multiplanetary. From the dawn of humanity, we have been explorers. Our ancestors traversed vast continents, sailed across unknown oceans, and looked up at the stars with a wonder and curiosity that's deeply embedded in our DNA. It's this spirit of exploration that has led to some of the most significant advancements in human history. Konstantin Tsiolkovsky, the early 20th century rocket scientist, once said, "Earth is the cradle of humanity, but one cannot live in a cradle forever." But the future of space exploration will rely not only on advancements in rocket engineering, but also on the mastery of biology. This synergy is vital for preparing humans and other life forms to survive the extreme challenges future explorers will have to overcome. Dr. Christopher Mason is the scientist leading the effort to make that happen. He's an investigator on many NASA missions and projects, including the famous twin study with astronauts Scott and Mark Kelly, as well as being part of the first team to sequence genes in space. He's currently collaborating with SpaceX to push the envelope of what's possible in space biology, plans to gather data from the upcoming Polaris Dawn and Artemis 3 missions. Most importantly, though, he is a big thinker and a dreamer with a plan. He's the author of The Next 500 Years, Engineering Life to Reach New Worlds, where he outlines a 10-phase plan rooted in biotechnology with the goal of making humankind a multiplanetary species. This plan includes everything from the initial steps of understanding and engineering our genomes for resilience to the long-term vision of settling new Earths. This plan is both ambitious and essential, reminding us that the drive to explore is not just about discovery, but our responsibility to preserve humanity. Dr. Mason, thank you so much for joining me.
Thank you, Tim. It's a pleasure to be here.
I'm so excited to jump in and delve into the 10-phase plan. But really, I think history will look back very fondly on this book as being a visionary blueprint that not only redefined our approach to genetic engineering and space exploration, but it also highlights the ethical responsibility we have to ensure the survival and flourishing of humans across the universe. So I wanted to start a conversation with a really fundamental question, and that is, why should humans devote tons of resources into becoming multiplanetary, and why did you dedicate your book to all humans and any extinction-aware sentience?
Great question. So I think it is, as outlined a bit in the book, also first, again, thanks for having me. A really pleasure to be here. I think the book was really a labor of love. It's actually, it was a joy to write because it's something I've been thinking about for a long time. And I dedicated it to I could have, at first I was thinking maybe I should dedicate this to all humans because humans, as far as we know, are the only species with awareness of extinction. But then the book, of course, is over several centuries. And I was thinking long term, we may not always be the only extinction aware species. And if we do things right, we definitely won't be. So I thought it could be for AI in the future. It could be for any sentience, any sort of aware species or entity that is thinking about the frailty of life and the fact that life is so transient, and then also can work to prevent extinction. So it's dedicated to current extinction-aware species, which is so far just humans, but importantly also to anything that may emerge in the future that could also have that capacity. And I think about this because life, so it assumes that life has value. Some people might say, well, I think nothing in life has value. I probably can't convince them of much of anything if someone is starting from that place. But if you just assume, two facts, one is that life has value and that the sun will engulf the earth, it necessitates thinking across a long time frame of protection of life. And if you realize it's all in jeopardy and that we can protect it, we can move it to other planets, well, then we should. It is something that is, if it has value and we can protect it, then we have a duty to do so. So I think I've been thinking for a long time about, the philosophy and the ethics as the why. And that's what kind of became the book.
And when did you start thinking about this book? Because you've obviously been practicing as a intellectual and academic for a long time now. When did you start actually thinking about this whole project?
Like really, I guess so kind of comically in middle school and a little bit early of high school. Wow. I actually did. My brother still makes fun of me for this. So in high school you can sometimes do what's called the Model United Nations or the Model UN. You have a chance to pretend like you're part of the United Nations. Or actually, we did one So we did the Model UN, and I also did one called the Model OAS, the Organization of American States, which is kind of like the United Nations of the Americas, North and South America. So I got Panama for my country, and this was in sophomore year of high school. And I thought, okay, well, Panama's not going to really build a space program. They don't have, the infrastructure. They don't have but I thought a lot about, going to space and thinking about ways to get there and support it. And I thought, but one of the problems spacecraft have is they don't often know where to land. They land in the ocean. Sometimes they land in Kazakhstan. But I thought, well, what if we had all the states of all the Americas make landing strips in each of their countries, so any spacecraft coming back from Earth would have more locations to land.
Right.
And so I wrote this called the Preservation of the Human Species resolution, and then introduced it into the group. And everyone said, "Who's this guy? Why are we talking about this?" Because they created like a fake like a crisis, like an embargo is happening between I think Costa Rica and one other country, and they made it a political crisis that the delegates had to address. And so my resolution did not make it out to the vote because everyone thought this was a little too out of relevance at the time. But I've been thinking about it ever since I was a kid, because I think it's really just those simple facts. As soon as I learned that the sun has a finite lifespan and it will engulf the Earth, I thought, that's a very clear end date that even if we have perfect world peace, we're all still on Earth, we will all perish. And so I thought, well, we should start to think about moving at some point. And so I've been thinking about it ever since I was a kid, ever since I learned about the normal life cycle of the sun, frankly.
That's very impressive for such a young gentleman at the time. No, I think what's really cool, what I thought a lot about, that was in the back of my mind when I was reading your book, was what I've always thought about is humanity's drive and necessity to explore, right? And to never stop exploring. And it seems that for a time, that flame kind of went out, right? And now with your approach and what Elon Musk is trying to do with being able to rapidly reuse rockets, I think that is now something that is really on the table. And I think we should never lose sight of that. Why explore space? There's nothing out there. We can't afford it. We can't go far. But if you let that flame go out, you're really never going to achieve anything. But also with that pressure that you mentioned, not only the burning out of the sun, but also heat death, which is, theoretical, but also very far into the future, but something as humans we should not, give up on. Mm-hmm. So I think why don't we start with sort of the first phase of your book, which is trying to learn as much as we can about the human genome. And you really kicked that off with your twin study with astronauts Mark and Scott Kelly. And there were some really fascinating results that you got from that. And I think, to me, as a molecular biologist, it was the fact that returning to gravity seemed to be tougher on Scott than it was going up in the first place. Lengthening of telomeres, genomic instability also reverted upon return, and that there were fewer mutations than you expected. Which of those do you think, or which of those do you find the most interesting?
I think as a geneticist, the telomeres was the most shocking and unexpected. Really just because we thought it would go the other direction, the stress on the body, the radiation. And so what's interesting, though, is if you look at the papers we published, the average does get longer, the average telomere length. And so the first question is really, the first question was, is that real? Like we thought, okay, does that make sense? So we did replicates across multiple labs, did different methods. We did qPCR, we did nanopore sequencing, did short read sequencing to look for motif differences. So looked at many ways to confirm it. And then once we did, we thought, okay, let's dig into that a bit deeper. And it turns out the average is longer, but as most of your audience will know, and you'll know, like the average could be a big distribution of high and low numbers. And so even if the average goes up, you look at the flow and the range of the distribution of all telomeres, which we can measure. And there's also more shorter telomeres. So even though the average is longer, there's also evidence of the damage from radiation with some shorter telomeres that you see more of them. Now you do see more longer fragments as well, and enough of them are on average longer. But if you look at sort of the shift of distribution, there is some evidence of radiation that's there in flight. But then when they get back, that's when we see the telomeres the average goes back down. And if anything, it goes a little bit lower than was pre-flight. So there's more short telomere fragments after flight. So even though in flight, it seems like there's two things we think happening is that when we brought it actually back to Earth and said, can we see this in other species? So we can see it actually in Plasmodium falciparum. We've published that. We can see it in cell culture, in vivo, in vitro. We can see it in both places. So if we just do radiation of cells, you'll start to see the same thing. So whereas if you take cells and simulate microgravity, and what are called these random positioning machines that try to simulate microgravity on Earth, we do not see it. So we think the biggest driver is radiation. And we just published a paper recently that showed it's a non-coding RNA called Tera that actually goes and binds to where the breaks are occurring. It's probably driving this telomere elongation. So we think it's the radiation that's driving some of these non-coding RNAs that are changing the telomere length. And it's probably also killing cells that were close to dying anyway, and then they'll just get shuffled away. And the other thing is, if you look long term, it is probably a little bit. You do see some evidence of the damage of the radiation with shorter telomeres when they get back. So it's a longer story. Like most things in biology, it's more complicated than just the headline. But I think it's more interesting because now we can see that this is a consistent feature of spaceflight. We've now seen across about 7 different missions, dozens of astronauts. We think this is one of the more consistent features of spaceflight overall, actually.
Now, in regards to the telomeres, what I thought was also really fascinating is that you saw that basically as soon as you started testing for it on day 14.
Yeah.
That is a very, what I think, or to me seems like a very quick response. So that radiation, that blast of radiation, which is not deadly, of course, not going to cause cancer in the short term, had that sort of effect very, very quickly. Do you have plans to test that even earlier in flight?
We do. And actually, we just published a work on the Inspiration 4 mission with SpaceX that flew Jared Isaacman and the whole 4 civilian crew mission. So there, even on day 2, we could see some of the telomere elongation, not as dramatic as by day 14. But we, from the data we just published, it looks like this happens very early in flight, actually. So as soon as you get into that higher radiation environment, which happens, of course, within a few hours, I think the body and the cells are reacting to it and responding. So it's actually one of the more immediate responses of spaceflight is some of these changes.
Now, speaking of papers that you published, I think it's important to appreciate you guys dropped, from my count, 18 papers on June 11th. Did I get that right? 18?
Yeah, from our group, yes, and across the whole consortium of other people with space biology, it was 44 total papers of everybody all coming together. Yes, it was a big release, just happened.
Super impressive, but also that goes to show how much data you guys were able to collect in such a relatively short period of time with Inspiration 4. I mean, some of that goes back to the twin study as well. You're using some data from there too. Tons of data, which is really, really cool. So with the data that you guys publish and everything that you've learned, what is being employed today already based on what you've learned in terms of spaceflight? Is there anything practically speaking you guys are employing in terms of prevention or even treatment on coming back?
Oh, great question. So some of the same measures we're going to continue, like we did in Twin Study, some of the SpaceX missions like Inspiration 4, Polaris Dawn, like telomeres, immune function, cytokines, microbiome. So some of the metrics are going to continue, but the interventions, a lot of the ones we flew in Inspiration 4 are going up for future flights. So the butterfly device for the ultrasound, where you can actually do imaging of the internal jugular vein, or you can look at the eye. We're going to apply that again and try and do other scans using ultrasound imaging of the body to do continued monitoring and potentially diagnostics. We're probably not going to have the crew diagnose themselves just yet, but all the imaging data can be relayed back to the ground crew, and flight surgeons can help. So that's something that we flew for the first time recently and is now going to be deployed on several upcoming missions, which is great. There's also a viral assay we flew that can look for viral reactivation. So that's one thing that is a lateral flow assay that we might be flying in one of the upcoming Polaris missions. And then also the sequencing in space. We have a new protocol for doing blood draw extraction, prep, and sequencing in flight, kind of like what we did with a biomolecular sequencer mission with Kate Rubins, where we flew it and sequenced DNA in space for the first time. It was back in 2016. But now we have newer methods, newer protocols that give us a lot more data. And so that's also something we're going to be flying for future missions. And that gives you a way to actually monitor, if there's an infection, if there's a viral reactivation, if there's a spike in sort of immune stress. These are things that you can monitor. And to your last question, the countermeasures, the interventions, that's what's on, I think the earliest stage of actual development, because we can now match people's genome profile to what pharmacological agents would work best. So what's called pharmacogenomics. Can we match the right drug to the right person, ideally at the right dose and the right time? And so we're just starting to do that now for crews. It'll become much more important as crews to go to the moon or eventually Mars, because you literally can't bring every drug with you.
Right.
And you also wouldn't want to. You want to make sure you only bring the drugs that would work on someone's genetic profile, and then you can manufacture, in situ in a new location, and then also be able to, because most drugs expire after 6 months or sometimes even faster.
So let me ask you about PCR, something that I did a lot of. And as soon as I was reading your book and also your papers, the first thing that struck me as somebody who really loves optimization, Is there anything, the first time you went to go do it, you might not have known, but is there anything different about PCR in space than there is on Earth? I mean, did you guys get a chance to optimize for that?
Good question. So mini PCR is the devices that we used for the amplification, and for a lot of the library prep. Some of the first libraries that we flew were already made on Earth and flew up to space, so it made it a little easier. Whether you've actually tethered the motor protein to the template and then run it through the nanopore sequencer. But for the PCR, not that I've seen, if anything, we had a joke that some of the very first data we got for the nanopore sequencing, we got better data in space than we did on Earth. Like the reads were longer, we got greater yield. So we were joking for a while that all the experiments should be done in space if you want to get the best quality data. But for PCR, to my knowledge, nothing ever looked worse. If anything, it looked better from some of the experiments we've run. But at this point, there have been probably a few hundred experiments with mini PCR on the space station. So I haven't looked at all of them, but from what we've seen, it's been as good or better, actually.
So there's because it's well known that microgravity environments do have an effect, especially on diffusion of molecules, right? So the fact that they might be better in space, that is very, very interesting. And I could imagine pipetting in space might be an issue too. I mean, there's a video of one of your colleagues on the zero-G plane, losing all his pipette tips. Which
yes, one of my favorite videos ever, actually. Yeah, Andy Feinberg losing all that. You should put it, I guess, in the link maybe for the podcast. But it is anyone who's done molecular biology at a bench, it is the funniest thing to watch because it's exactly what. So that's why when we fly pipette tips, we don't fly them in the racks. You'll see that video. You'll see why. But instead, they're more in small containers, more tubes that we can actually get up there.
Right. Yeah. I mean, we've all knocked over, a rack, but imagining that in space, incredible. Now, I guess, if we look at your 10-phase plan, we're really out of phase 1. We're really now in phase 2, which is essentially the preliminary engineering of genomes. You've got that slated as being between 2021 and 2040. So on a timeline, you're correct in terms of the timeline, in terms of what we've achieved or you've achieved so far. I think we're right there. So projecting short term, how do we go from, I guess, first of all, a little bit of detail on phase 2, but how do we move into phase 3 with the long-term trials of human engineering that's required for that 2041 to 2100 timeline that you've got set up?
So this is where we're in the wonderful early days where we are now doing CRISPR of somatic cells to treat disease. There have already been CRISPRed human embryos that have been made but not grown to term. There have been, of course, the CRISPR babies that were born through a pretty shady process, but still, it's happened. So a lot of things that were, hypothetical only 10 years ago are now already occurring, or even just standard in the case for beta thalassemia or some sickle cell disease cures. And I don't know if it's of course in the book, but in the audience, I think one of the most extraordinary things is it's not actually just about swapping out one part of a gene or another. It's actually reactivating fetal hemoglobin to cure the problem with adult hemoglobin. So in all of our bodies, of course, we have tens of thousands of genes, but some of them get turned off. Many of them get turned off in many cells, but fetal hemoglobin, as the name implies, is only active when you're a fetus, and then it goes away. But the gene is still inside of us and can just be reactivated, in this case as a cure, which is extraordinary. So I think the genome engineering trials I've anticipated would be, again, I wrote this first draft in 2010, so I didn't quite know what would come. And when I first wrote that down, CRISPR was not really well known yet. It was in some of the literature. There were talons and there were zinc finger nucleases that were coming online, but I never thought it would be this easy. So if anything, I thought I was being more lenient and optimistic with my projections of when we start doing the genome engineering. But we're already there. And so what's extraordinary is the new CRISPR systems or prime editing systems enable very exquisite editing of different loci across the genome or activation of older genes that have been turned off. And so I think the first generation is happening. But what I wanted to start thinking about is monitoring people over many years or even multiple generations, much like the Framingham Heart Study did, where we've actually monitored people over a long period, because then we can start to discern, are there other issues that came up in the editing that we couldn't yet see, or there maybe there's off-target effects is one of the biggest terms, where just things have been edited that you didn't initially anticipate, but that can happen, and of course does happen. But I think also, are there just are edits incomplete, or did we not understand how there are gene-gene interactions that we just didn't quite appreciate yet? So a lot of that is in this next, well, frankly, the next 15 years, we'll have a lot more of this kind of first testing And then the goal is by 2040, 2050, it becomes more routine, and then we start to do the intergenerational kind of studies.
So how much of the genetic engineering that you speak of in your book do you foresee being sort of somatic engineering and then embryonic engineering? Because some of the problems that need to be solved that I assume you're referring to have to be done almost on a embryonic level. So it's almost like you're projecting for the future of astronauts. Are we breeding astronauts in advance of space flight? That might be a thing a couple of 100 years from now, but how do you see the split?
Yeah, so if we get good enough at somatic editing, so just also to clarify for the audience, the germline would mean all the cells are changing, including the sperm and egg, and it gets passed on to the next generation, whereas somatic editing is only for that one person. If we get good enough at somatic editing and you can reach all the cells in the body, which again, we can't yet, but in 20 years from now, maybe we could, you may not ever need to do germline editing. So I think a lot of the technology and the focus is on somatic editing. Of course, as you well know, the challenge is getting to each of the cell types. It's much easier to edit a few hundred cells instead of trillions of cells, tens of trillions of cells, just by the numbers game alone. It is harder. So, and delivery is obviously the biggest problem to get to this right tissue, the right cell type, at the right time. So I think that is one of the biggest challenges, but I think it's been getting better and probably will continue to do so, where we can map specific cells that are the most responsive, are the most amenable to being edited with the right construct. So it'll take some time, but I think that's where that gets more into kind of the phase 3 area of the plan.
And one thing that I was thinking is in terms of a hurdle when it comes to somatic editing and trying to hit every cell or all the cells you want to hit is the fact that people don't really consider that we're really a genetic mosaic. Not every cell has the same genome, despite what you read in textbooks or introductions to textbooks. Yeah. So that would be something that needs to be overcome too. Mutations that you pick up over the lifetime of a cell get spread out through the body, and not every cell is the same. Yeah. Right. So phase 3. So really we don't have to go through every single phase. I encourage everybody to read the book because it's a fascinating, one of my favorite nonfiction books ever, especially because it brings together two of my favorite topics, genetics and spaceflight. Being a spaceflight nerd, which I'm assuming you are too. Yes, sure you are. My question is going to be, what's the end game? Where do you see, let's say, phase 8, 9, and 10? What is the ultimate goal? We're trying to become multi-planetary, but how do you see that play out? It's hard to say practically speaking because we're not there with the spaceflight technology. But what do you imagine being sort of the way we do that?
Yeah, we can jump to the other phases. A lot of it's about finding new tools for synthetic biology or blending organelles and genes between species, some of which we've published from my lab, and many people have done this as well. But also, the other phases are just finding can we get to Titan? Can we get to other planets that could support different kinds of life, and looking at extremophiles. But then eventually, towards the later phases, that's where I think we get into these fascinating concepts of generation shifts, where we can, this will be then 200 years from now, where we could, and I put examples of planets that are already in the book that, I knew when I first published it, a couple of years ago, really great exoplanets that if we could get there, we could possibly even survive today. They may have liquid water. They'd be in the Goldilocks zone of not too hot, not too cold. So I think there's really a great planets that we could try and get to today. And so the challenge is propulsion. We can't get there that quickly. And also the radiation in interplanetary and interstellar space would be pretty high, but not necessarily impossible to survive. So we think, there's and that's where it gets into the question of, should we bring in radioprotective mechanisms from other species to engineer astronauts to be able to make the trip? And I think maybe we'll be wrong. Maybe the human body is more resilient than we think. Maybe we can do combined humans plus some robot missions. But at the end of the day, unless the robots can carry all this sort of duty with them to protect life, which again, maybe they can, but for now, we don't think they can. It is, humans will probably have to go, and eventually, it's the only way we'll survive the death of our sun. So the later phase is called the launch toward the second sun. The goal would be that we have enough knowledge of radiation, genome engineering, spaceflight, and propulsion to make a generation ship that then gets to one of these other exoplanets that we could likely survive on. And then that brings in all sorts of questions. Like if you were born on the third generation of this ship and you know that your whole life you'll live and die on this one spacecraft, some people would consider that prison. Like you're marooning this ecosystem and the subset of people to live and die on one small ship. And I always like to say, well, it's just a difference of scale. Like right now, we're all stuck on one ship called Earth, and you can't go to any other place. Earth is beautiful and wonderful, and I don't want to discount it, of course, but it is still just a big spaceship. It's very big, but it's still, we can't go anywhere else. There's not another, can't yet go to Mars and back. And so I think this, it seems like an odd framework, but it's something we should seriously consider. And I presume no advances in propulsion from my estimates in the book.
Right.
That we will, but the people, NASA engineers and others are working on fusion drives. People are trying all sorts of ideas as we speak. So I think, yeah, but none of them would make it so we're, traveling. Yeah, well, even if it was at light speed, which of course won't happen, it would still be a long travel. It would be many, many years and probably still involve multiple generations, even if we get to fractions of light speed. So that's why I think about what that would be like on the ship. How do you engineer a society that views themselves kind of as the almost like the chosen ones that are going to carry the torch of humanity and life into a new solar system?
Let me ask you a fun question. What do you think we'll hit first? Phase 10 or Kardashev scale 1?
Yeah. Well, phase 10, the last phase is like, if the universe ends, at the heat death of the universe. So you mean like between the heat death and
No, I mean your 10th part of your 10 phase plan is sort of 500 years from now. Do you think we'll get there first, or do you think we'll become a Kardashev scale 1 sort of society where we have control of all sort of energy on Earth
and the sun? I think Kardashev scale 1 is you also can control all the sun's energy, right? If I remember right,
I think that's 2 or halfway. 2. Yeah, that's 2.
You control. I think probably we'll be able to launch generation ships before we could control all the energy on the planet. Kardashev level 1, I think. It is a fascinating concept, and I hope. I mean, that would be, or things like a Dyson sphere where you can capture all the energy of the sun, which I think is level 2. Extraordinary feats of engineering, but and they seem almost impossible. But so many of the things that we do today seem impossible to people that were alive 100 years ago, or even especially 200 years ago. So that was what was kind of fun about writing the book, because I kept doing the inverse. I'd say, okay, what did people think about 100 years ago, or 200 years ago, or 500? And to your point in the beginning, people were still explorers back in the 1400s, people going on ships that were going across the ocean, and many of them just did not come back. They would say, I think I'm going to come back, but I might not, right? And the drive to explore, sometimes people were avoiding persecution, of course, which is not ideal, but some people were really just wanting to explore, either for treasure or for curiosity. But it really was, that drive has been consistent throughout all of human history, but the technologies has changed, of course.
Now, to your point, where you're talking about, a couple hundred years ago, or now we're doing things we couldn't even imagine. I mean, sequencing is developing faster than Moore's law, which is something that is just incredible. So what we can do, another 100 years from now in terms of molecular biology, who knows? We were talking about propulsion and engineering, but on a genetic level. So let me ask you this question: what do you think space exploration would look like in the absence of genetic engineering? What do you think the limit is?
Yeah, if you just send people, I mean, because we've been sending bacteria and plants and humans up into low Earth orbit. In the book, I describe this a bit as the, send people and hope method, where you like, well, we hope you survive and bury under, it'll obviously be dark. You'd be spending a lot of time in caves, which you might do anyway, but I think you would have to be protected from the radiation. But there is a chance that if it's been 200 years of humans living on Mars, for example, that new population would be strongly selected for those that could survive more radiation. And we already know from current data that there's a wide range of response to radiation from just the normal population that exists today. Some are more sensitive, some are less sensitive to ionizing radiation. So you can imagine a case where you're selecting for over many generations the people that would live on Mars that could tolerate radiation and would slowly get closer to the surface. So I think a lot of the changes genetically might happen over time, just through evolution, but the engineering would let us move faster and get to the desired goal of really sustainability much quicker.
As they say, hope is not a plan. So let's use the 10-phase plan. Now, you mentioned synthetic biology and genetic engineering. A lot of it is in terms of human genetic engineering, but you also touch upon being able to engineer other organisms to generate food, recycle waste, et cetera, et cetera. Could you speak to that and a little bit about the extreme microbiome or your XMP project?
Yep. So we have a few projects to try to learn from extremophiles, and I think I've described human genetic engineering, and CRISPR has been in the news, and most people are probably familiar with that now. Actually, even my daughter just graduated from middle school, And the whole class knows that, CRISPR is just a verb that they routinely throw around. So it's really an extraordinary time in which to live, where children just presume routine and simple genome editing, with no concept of how hard that used to be or even impossible, right? So it's actually really, to them, it just seems like, well, of course you'd edit genomes if you need to. Like they don't even question it. So it's really this interesting, bifurcation of people that have grown up with it or been in the science field versus those that are just frankly older, who to them didn't even know it was possible. And so, for example, my grandparents or they just, to them, it seems like it doesn't even make sense, right? So anyway, the engineering those for more than humans, and indeed, you could do this for synthetic biology. You could make modified cyanobacteria. You can mix and match genes between, say, tardigrades and human cells. This is the water bears that can survive in space. I talk a lot about biomanufacturing. So ideally, you'd have automated systems that are probably in yeast or other organisms like E. coli that can have modified genetic code or make the pharmaceuticals you'd need or make the food you would need, because you'll need simple structures to be able to just survive. There won't be many raw resources on the moon or Mars, so you'd have to recycle as much as you possibly could. And again, you ideally could take systems that work today, but you may have to engineer them to be much more Spartan and use every bit of every atom you can get when you're recycling it from a space station or from a new settlement on a planet. And then the XMP is a project called the Extreme Microbiome Project, where we try to map adaptations from anything we found that seems to survive in a high radiation environment, high salt, essentially high pH, high or low pH. These really extreme organisms, which are called extremophiles, that are microbes that have managed to survive and live in boiling water. For example, TAC, where we get TAC polymerases from, an extremophile. So we get these organisms that show us clues on how to adapt in what would seem to be impossible conditions, but then those can help us. Basically, I like to say it's a molecular library of life, where if we get all the adaptations from all the organisms on Earth, that gives us basically a large catalog of tools to deploy for surviving on other planets.
Now, another thing I really liked about your book is the fact that you don't touch upon just, engineering, genetic engineering, is that you've got some philosophy in there too, some interesting points to think about. And as I was reading it, I had a conflicting sort of idea in my head, is that, is it important to remember our humanity, right? Since we're trying to preserve humanity, that's sort of the goal that we're talking about here. Is it important to remember our humanity when we are using genetic engineering to preserve it? There's sort of a, there might be an argument that once you start engineering people on a genetic level, they're no longer people, or, there are some extreme views on that. What is your view on that? Should we remember our humanity when we're trying to preserve it?
Yeah, I think we should, and it's all, I think it's you don't have to do one or the other. We can, advance humanity into a distinct state, as well as remember our ancestral states. So I think the best analogy for this would be, we still have genes from Neanderthals literally embedded in our genome. We can't hang out with Neanderthals; they're all extinct. But we still have bits of them inside of us, and that influence some of our traits today. And I think in the future, though, we'll have a lot of communication between the planets. And so you could imagine, let's say the people on Mars become so dramatically divergent that they can't even come back to Earth, which I would consider a failure of engineering. We should make it so that people could go back and forth. What I call planetary liberty in the book is that you should be able to go to and from each planet at whim, and that would be a good success of biological engineering. But if you couldn't, and let's say that Mars was just too distinctive an environment and they really became a new branch of humanity, I think that would be fine. The same way, if you have kids and they go off to college and they build a lone life in a different country, and they build an entire world and new friends and have their own kids, it's a good thing. It is, as long as they don't turn back and then try to start a war with us, that would be bad. But I think short of extermination or strive for war would, which has happened a lot in human history, but hopefully that doesn't happen here. I would view it as a good thing. It's another shot on goal for evolution, for life to persist.
I'm glad you brought up planetary liberty. If I may, a quote from your book. The ideas in this book are not simply to prolong the survival of humans, but also to achieve cellular, molecular, and planetary liberty. This dream is not only for humans millions of years in the future, but for all species from any kind of matter. What is planetary liberty? What's cellular liberty?
Yes. So I think, bringing up some of the points you just did of somatic editing and getting really good at genome editing. What's extraordinary is you could imagine a state where when you're born with a disease that you in your genome, like Huntington's disease, or risk for ALS, or it could be BRCA1 mutations for breast cancer. Whatever, historically, the genetic shuffle you get at birth is what you have to deal with, and there's not much you can really do about it. But in the near future, or even to some degree today, you are not beholden to the genetic deck you got at birth. You can change the context. You can change actually the structure and the text itself of the book of your own life. And so that already means we have to some degree cellular liberty, that you can do what you want with your own cells. You can modify the genome, change their states. And then eventually I think of the liberty of a personal liberty, where you can do what you want, walking down the street and you can decide to go get ice cream or go get a bourbon. You can have the liberty to do what you want. You have free speech. You have these freedoms that we take, for granted a lot in the United States, but are in most of the other parts of the world as well. But long term, the planetary liberty, like in a true sense of like liberty, is the ability to have freedom in your choices, and also security and safety in your belonging, kind of like Maslow's hierarchy of needs. But the liberty is the ability to go places and to do things and to have freedom of your choice and expression and speech. And planetary liberty is just like the fact that your physical body could survive on more than one planet, because right now we definitely don't have that. We can survive on Earth for a while, but we certainly, without structures, could barely survive. We couldn't survive on any other planet without, a spacesuit, and even that short term. So the planetary liberty, the dream would be that, as I alluded to before, is that you can go. Maybe you may have to do some somatic editing to say, okay, you're going to go. You've been living on Mars for 20 years. You'd like to go visit your parents back on Earth. But you need to do 6 months of somatic editing to prepare your body to get back, would be something we should consider. But I think and again, maybe I'm being too conservative. Maybe the human body has extraordinary plasticity and we might not need to do that. I would take that as a win. But we have to at least consider what we'd need to do to make it so you could survive back and forth between planets. And if you can do that, then it means the same way we take it for granted that you can get on a plane and go to Copenhagen or go to Tokyo and just go visit another place. We could do that then between planets. And so I think the same way we think of country, the liberty to go visit another country, we could do that with planets and do that with your body. So, which I think which I'd want to endeavor to create, because otherwise you would then be stuck on one planet. And I think that I'd like to have the ability to go to more than one planet if I could.
Yes, this pale blue dot is beautiful, but
Yes.
let's not stop exploring. Now, to my knowledge, correct me if I'm wrong, I don't think anybody's done the sort of molecular work that you're doing on anybody who's ever done an extravehicular walk or, in space in a spacesuit. Do you have any plans in being part of what SpaceX is going to be doing with Polaris Dawn coming up?
Yeah, absolutely. Yeah. So, and the mission should launch, if all goes well, next month. And it'll be, so we're doing the blood work before and after that spacewalk. So it should be really fascinating to see how does the body respond to, of course, they're not in the vacuum of space. They will be in their spacesuit, but this will be really extraordinary. You're right. To my knowledge, also, no one's done a pre and post spacewalk, because it's pressure on the body. It is very stressful on the body. We had a call this morning in lab where actually we're going to do for the New York City Marathon this November, try and get blood from people during the marathon. It's starting to look at more conditions where the human body is under extreme stress or duress to build a better context for it. Because a lot of times people say, well, is a spacewalk like scuba diving, or is being in space like climbing a mountain? And so we think the answer is partially yes. Like, for example, telomeres did get longer when we took Mount Everest climbers, for example. So we do know that, but we want to build some of these better sort of molecular maps of what happens to the body in these sort of extreme states.
And what about, stop me if this is confidential, Artemis 3, you going to be a part of that?
So we'll, Artemis 2, I think as most of the audience probably knows, we'll just go around the moon and back. Artemis 3, there'll going to be some life support systems that we're working on, but the actual blood draws, if all goes well, yes, we'll actually look at telomere changes for Artemis 3. And it's some of the final flight plans, of course, have not been announced or finalized. But if all goes according to plan, yes, there's a study that NASA leads called CIPHER, which is an integrated portrait of a lot of their astronauts. And it's based on a lot of the work in the twin study. So that we'll be doing not everything we did in the twin study, but as much as possible that we can for that mission. Also due to some of those crews. And so it should be the first person to color on the moon, plus some of the first molecular data of someone who's been on the moon. And we just recently got yeast that flew around the moon in Artemis 1. So we did the genomics of that sample and just submitted a paper on that as well. So I think we'll see more about that in the coming year.
Oh, that's exciting. I'm looking forward to that. Now, something else that you speak of, we talk a lot about going away, but coming back in terms of planetary protection. Can you talk a bit about that? Because there's going to be a chance 2033 some rocks from Mars will be coming back. What is the idea behind planetary protection?
Yes. So planetary protection, for those who don't know, is the idea that you want to avoid forward contamination and reverse contamination. Forward is we bring something, say, to Mars and we contaminate the planet, or we bring much like what some of the settlers from Europe did when they came to North America and they brought smallpox with them. So we want to make sure that we don't bring a contagion or a really threat to life that might be on Mars. So that's forward contamination. Reverse contamination is if we bring something back, which is supposed to happen in about 2033. That the Mars rock has the Andromeda strain and kills everyone on it would also be pretty bad. So
great book on Michael Crichton.
Yeah, exactly. Which it was a good book, but actually he noted when he first proposed it to publishers, they thought it sounded too outlandish, but eventually it was very well received.
Right.
And it could, it's something that there's supposed to be a BSL level 4 facility and then something contained around that for when samples do get brought back. And, we've also been working with the JPL in the clean rooms where they build the spacecraft, including the 2020 rover that went up, which now, in Resilience that went up there, sorry, Perseverance that went up there. We have been profiling the microbiome of the spacecraft and the room where they get built, so we can have a backlog and really a baseline profile of what the room looked like when they built the spacecraft. So if we do see any nucleic acids at all, we'll be able to quickly compare anything that's ever been observed in that clean room just to look for contaminants. But if we think we find life geochemically, molecularly, and some trace elements, then it would be pretty exciting for sure.
Another fun question. I'm assuming that you're a fan of sci-fi. There's no way you're not.
Definitely.
Is any movies out there or anything that you've seen that sort of matches your percent or your vision of the future? What's your favorite sci-fi movie? In terms of just favorite, but sort of that might match what we're looking at here.
Yeah, I guess a handful of maybe some favorites. Like, of course, Isaac Asimov's Foundation series was one of the most formative books I've read as a kid. There is, even I think Interstellar as a movie is a fabulous movie that has a real sense of the drive to preserve humanity and ways we could get to other planets. I recently read a book called In Ascension, which just came out last year, which is about sort of humans trying to go out and explore past the heliopause, which is a very good fiction book, also about some of the radiation in deep space and how you might deal with it. And then, I think, of course, I watched Star Trek, Star Wars when I was a kid and saw those movies, but I think Kim Stanley Robinson's books on Red Mars, Green Mars, Blue Mars, of course, were great. And even Seven Eves by Neil Stephenson, some recent books I've read, I guess. But those, the one I think, the one that actually was really intriguing to me was The Expanse, the series of books, also the series on TV. And what is fascinating to me about those books is it has this great assumption that humans can quickly move in and out of the solar system and eventually go to other solar systems, which is wonderful to think about technologically. But then the thing that makes me sad about that series, although it's very entertaining, is that even though humans in that series, in that realm, have conquered the solar system and we can go anywhere almost at whim, we're still just as tribal and backstabbing and petty as we are today. It's like, there's war, there's conflict, there's people theft. And so in my mind, I would hope that we'd reach some sort of more utopian Star Trek universe once we're, jetting around the stars. But those books in that series might be more reflective of what it would be like, much to my chagrin, is that we might still be just, people fall in love and people get angry and people get jealous and people are imperfect. So, and petty sometimes, right? So, and violent, the wars, there's a war between Mars and Earth in that series. So I think I love that series, but it also saddens me that we would reach that stage and still be the same sort of tribal entities as we are today, just between planets.
Yeah, some things never change, and some things genetic engineering might not be able to change as well.
Yeah. Well, I've had this question before, could you genetically engineer, get all the violence out of the genome or something like that? And we do know the things that increase or decrease tendencies. So, in theory, you could imagine a pacifist planet that you've made these really peaceful people. Which itself is a weird question. Should we do that? I don't think we should, but it's interesting to think about.
Now, if you don't mind a personal question, what motivates you? I mean, you've got a lot of work on your plate. I've got a couple other things that you work on I want to ask you about before we wrap up. But what motivates you as a scientist, as a human, as a father?
Yeah, as a father, husband, as a human, I'd say, actually, it still is one of the things I think about almost every single day. It's just the simple fact that the sun will engulf the planet. And so, but I don't think it doesn't make me afraid. It just makes me motivated. It just thinks that I take great joy in sort of the splendor of a summer day, of seeing children play in the park, of seeing art and seeing music created, seeing all the science that humans are building in architecture and even in politics, and all the complexity and nuance and beauty of what humans are building. I take great comfort and joy in it. So I wanted to persist, right? So I think this, and I think to our knowledge, it is unique in the entire universe, life itself. I mean, I'm hoping we're wrong. Maybe we'll be wrong, but also about 90% of all the stars that have ever formed in the universe have already formed. And so I think, one of the questions of like, where is everyone, which Fermi said, is there the great filter that's preventing us from talking to other civilizations? There's a chance that maybe a lot of them have already come or gone, or maybe we won't find them, or they'll be too far away. So I think it is very precious. So what drives me a lot is this, is the preciousness of what life as we know it, and also the joy of discovery. Sometimes just even going from the subway to lab, it takes too long to walk, so I will just start to run because it's taking too long. So I'm very blessed and honored I get to do what I do.
Now, in terms of responsibility or to preserve humans, something else that you're a part of as an advisory role is the Colossus project, which is the efforts to de-extinct, in this case, the woolly mammoth. Mm-hmm. What are your views on that? How far do you think we are on that? Do you think we should? Clearly, you're a part of the group. Do you think we should? Can you tell us a little bit about that project?
Sure. So I think we should, because in some cases we killed the species, we made them go extinct. And so the analogy there would be, if you murdered someone, but you could unmurder them, would you do it? And most people, I think, would, unless you're a psychopath, maybe not. But there are things like the dodo, there are species that we've probably helped make extinct or directly made go extinct. We can bring them back, but also the tools for conservation that are being built by the company. Again, full conflict of interest. I'm on their board and I'm an advisor, so I know a lot about the company, but I really enjoy what they're doing and how they're doing it, and the drive to improve how we monitor life, protect life, and then even engineer life to save life. So I think these are the technologies that we'll need to potentially help us get to other planets, but also to preserve life as we know it. And it is very fragile. And some people say, well, 99% of all the species that have ever lived are all gone, so what's so special about this time? But I think what's special is there's never been a time where a species existed that could understand that. And again, that's just us so far, and then do something about it, right? So we're not just passive observers of the frailty of life and extinction. We can actually prevent it. We can even reverse it. So I think it's an extraordinary pivot point of humans' capacity and also responsibility. So I'm very glad to be a part of it and help as much as I can.
Very interesting. Right. We'll see how that goes.
Yes, it will be hard. It is definitely going to be hard. And the woolly mammoth is one. The dodo's been announced. The thylacine. So there are species already announced and several others in the works. But it's going to be hard. But it is it's great though that it forces us to better understand the methods of multiplex genome editing, genome restructuring, genotype to phenotype maps. All these fundamental questions in biology. It helps us understand them better.
All right, two more questions. One nerdy, one not so nerdy. How much have you thought about, because you talk about biosignatures and life signatures, how much have you thought about the potential of different nucleotides existing out there in different forms, maybe more than just the four that we know of in the genome, at least, and the possibilities that might render?
I think about it a lot. As a section in one of the later chapters about it in the next 500 years, it's almost certain if there are if nucleotides are what the stuff of life is on a different planet, they would almost certainly look different because I think the odds of finding this exact conformation again on another planet would be, my guess would be quite rare based just on, but you would need to have some pairing mechanism. You could have, instead of 4 bases, you could have 6, instead of 20 amino acids, you could have 30, or you could have, 16, you could have some variation for sure in the basic counting units of life. And I would be surprised if it was the same. It would be extraordinary evidence of panspermia if it was the same. That's, life has just been essentially like a shuttle between planets, but it maybe only formed once. So that would be fascinating if we found life in 5 other planets, let's say 1,000 years from now, and it all looks the same. That would actually be pretty interesting evidence of a common origin, which would be fascinating.
That would definitely be fascinating. Now, sort of the goal with this podcast, personally for me, is to motivate young scientists, to expose them to what's possible out there in terms of careers and possibilities. Do you have some words of wisdom for anybody who's in college and university getting ready to sort of get out there in the real world? some words of wisdom, advice?
Sure. I think a lot of it is to, don't ever, give up. A lot of time, and I mean, from the simple thing to I emailed the professor, but he or she didn't email me back, or then send it again, or I submitted a paper, it got rejected. You submitted a grant, it get rejected. That failure is inexorably essential and critical part of the whole process of science. That experiments fail, grants get rejected, papers get denied. You not everything works the first time. Very rarely do things work the first time. So I think knowing that, and knowing that if something fails, it doesn't mean that you're a failure. It means the experiment failed. Now, if you do the same experiment 5 times and it always fails for the same reason because you didn't have the right enzyme, well, then maybe you're contributing to that failure. But at least the differentiating from what is a failed experiment versus a failed person. And very rarely should you ever think, oh, I'm the failure because my experiment failed. Is really or that I'm the failure because this person didn't email me back, or because someone didn't, return my call. Like the only way something really has ever gotten done is just this continued tenacity and determination. And sometimes just even like volunteering. Like some of the best people I've had join my lab just emailed me out of the blue. And I said, oh, we're really busy, because everyone's always busy. And I said, well, but why don't you just join this meeting? And then it's just about like being persistent. Send a summary of an analysis that you did of some data that the lab already published, or say, oh, I read the last 4 papers from your lab and I found these interesting new directions, and just propose interesting experiments, or start like a dialogue, and be curious, and also just be useful. Sounds like a weird thing to say, but by useful, I mean, download some of the data and reprocess it. Get familiar with computational biology is probably one of the biggest tips. They'd be very adept at navigating and manipulating large data sets, because the biology and genetics and most of the fields are very data intensive these days. So that means to be useful, you have to be data savvy. And so getting very comfortable with coding, and also sharing your code and sharing the methods and being very transparent with what you're doing and how you're doing it helps reproducibility, but which is often, though, the opposite of what people feel like they have to do. They feel like, no, I need to hide my code and I have to just be the one person in the world that knows how to do something, because then you'll have value. But it's actually usually the inverse. If you show extreme transparency and openness to work and collaborate, people then will start to flock towards you and say, okay, great, this person is very helpful, useful, friendly, collaborative. So those are all good things I've found in the field. So be curious, be tenacious, be useful, I guess, be it in those specific ways I just mentioned. Yeah.
That's fantastic. I really appreciate that. Dr. Christopher Mason, author of The Next 500 Years. I highly recommend it. Thank you for your time, sir, and good luck with everything upcoming. We'll be following your work.
Definitely, yeah. Look forward to it. It should be a good summer and the next couple of years. And also, one last tip: if people want to get involved with NASA, the Gene Lab, a lot of the data sets are all available. They're all online. The code. Just start playing with the data. So NASA's Gene Lab, or the open science data repository, is where a lot of the data is, and it's a great resource. So if anyone wants to start getting involved in spaceflight, space genetics, jump right there. Feel free to reach out. And thanks so much for having me.
Fantastic. We'll have that posted in the show notes. Okay. Thanks so much.
Thanks.